Hello, my name is Ariely Barbosa Leite. I'm studying pharmacy at UNIFESP here in Brazil, and I'm doing my scientific internship in the Laboratory of Molecular Biology of Pathogens under supervision of Professor Nilmar Moretti. The main focus of my work was to study the effect of acetylation and the regulation of a glycolytic enzyme in Trypanosoma brucei. So what is acetylation? Acetylation is a post-translational modification that introduces an acetyl group, here showed in green, to an amino acid, generally lysines. These modifications can neutralize the positive charge of lysines, change its overall size, alters local hydrophobicity, and these changes can alter too the properties of proteins and enzymes. And what about our organism? The Trypanosoma brucei is an unicellular protozoan parasite that colonizes blood, skin, adipose tissue, and also brain tissue. And when that happens, the patient can develop the characteristic symptoms of this disease that is called sleeping sickness. These parasites, as we can see through the life cycle in this slide, they pass through different hosts facing hard environmental changes. So they have to adapt their genetic expression and enzymatic activity. And the main study forms of these parasites are the procyclic form in the invertebrated host and the trypanosoma long slender in the mammalian host that is also called bloodstream form. The procyclic form that lives in the tsetse fly, they found lower levels of glucose to produce energy, so they rely on oxidative phosphorylation. However, the bloodstream form that lives in the mammal host finds high levels of glucose and can use glycolysis as main energy source. These two forms were used in a recently paper published by our lab when we described the acetylome, that is the set of acetylated proteins, of some protozoas comparing the procyclic form present in the invertebrated host and the bloodstream form present in the mammalian host. And we found that these forms have different levels of acetylation. We found 288 acetylated sites and 210 proteins acetylated for the procyclic form. And for the bloodstream form, we found 318 sites acetylated in 285 proteins. But between them, just 45 were common among these forms, reflecting the difference in the mechanisms of adaptation of T. brucei during its life cycle. And the difference in the acetylation was even more evident in the glycolytic enzymes. So here we have a scheme of the glycolytic pathway, and we can see these yellow circles. They are present in different numbers between the enzymes of procyclic in blue and bloodstream in green. And through this heat map, we can confirm that lower levels were detected in the bloodstream form, which uses glycolysis as main energy source, compared to the procyclic form, which relies on oxidative phosphorylation for the ATP production. This found suggests that acetylation could play a role in regulating the activity of glycolytic enzymes in T. brucei, as previously showed for aldolase and glycerol triphosphate dehydrogenase in mammals. Landby and his colleagues have shown that when we change the lysine for glutamine in the position 147 in aldolase, the enzymatic activity approaches zero. And we decided to compare the structure of these enzymes, and we can see that the human aldolase and the Trypanosoma brucei aldolase are very similar. And making an alignment of these proteins, we can see that these residues marked here in blue that makes up the catalytic site, they are conserved, not only between human and brucei, but also in other trypanosomatids. And the residues are just changed the number for the position, but they are conserved. So to verify if these residues could play a role in enzyme activity, we generated mutated versions of parasite proteins when we replaced the lysine for a glutamine that mimics an acetylated lysine. We designed two strategies to obtain the mutated aldolase for the homolog human 157 and also for residue 117 and 163, which we detected acetylated in our analysis, and they are located close to the catalytic site of this enzyme. So the first strategy was using the same approach of Lundby, changing lysine for glutamine, and to do that we have used two sets of primers containing the substitution, and we have obtained these two fragments, and through another PCR reaction we obtained the complete fragment with 1 kb, and this fragment were double digested with restriction enzymes and cloned into the vector PET28A, and after the plasmids were transformed into competent cells confirmed by colony PCR and sequencing. Once confirmed, we used BL21 cells to express heterologous protein that was purified using a nickel column, and here we can see that the protein was eluting the soluble fraction and with around 41 kilodaltons. With these proteins in hand, we have used a colorimetric assay to test the in vitro activity for the native, aldolase, and the mutated. And we can observe that when we have a glutamine in the position 157 here in green, we have a high decrease of its activity, as happens in human. So we wanted to gain more insight of what could be happening in this enzyme. So using in silico analysis, we tested the electrostatic surface potential through Coulombic surface coloring on chimera, and we have compared the native with the mutated ones, and we can see that the substitution of glutamine in the position 157 shifts the electrostatic surface potential for a more negative state when compared to the native one. We also tested the binding with the substrate, that is fructose, and this test reveals that significant change of the way the substrates interact with the catalytic site of this enzyme. So our colleagues from UNIFESP executed some molecular dynamics to see the possible structures modifications during the time of the analysis. And here we have a 3D protein structure of native form with the pocket showed here in the blue mesh. And when we have this lysine acetylated in the position 157, we have this green mesh showing a reduction of the volume of this catalytic site. And through this graphical representation of this molecular dynamics triplicate, we can see that the catalytic site volume is highly reduced at the most percentage of the time here when we have this version acetylated. So these results and others that I have done were published this year in Biochemical Journal, and you can find the link for the paper in the description. Now passing to our second strategy, that was to replace lysine residues by a number codon to obtain the aldolase using a non-canonical amino acid strategy. This technique was described by Newman and his colleagues, where we have a cell expressing a tRNA and a synthetase, which was modified from anarchia, and this tRNA and synthetase can recognize an acetyl-lysine. So in the final process, we have the cell expressing the acetylated protein. In order to achieve that, we ordered this plasmid from SnapGene that contains the tRNA and synthetase to recognize the acetyl-lysine, and we are waiting the return of activities to continue the steps of expression, purification, and activity testing. So in conclusion, I will express this aldolase with amber codon for greater robustness of the residues that we have published, when we show that the investigated residues can alter the enzymatic activity of aldolase in Trypanosoma brucei, and the 157 seems to be the regulator. And for the first time, we showed that acetylation is a new regulatory mechanism of glycolytic pathway in this organism. I would like to thank my lab colleagues from LBMP, our other collaborators from UNIFESP, UNESP, and UNICAMP, and the founding agencies CNPq and FAPESP for the support, and also thanks to ISAP and ISOP for this opportunity to present my work, and thank you for your attention.